has it started yet? Okay. So we'll be talking about another hallmark today, that is loss of proteostasis. Anybody could tell me what is proteostasis?
Maintenance of the proteome.
That is correct. That is absolutely correct. So what happens when the proteostasis machinery is lost? Then we have accumulation of damaged proteins. And there are three interlinked arms of this proteostasis network, which involves initially the protein synthesis and folding. Then there is the conformational maintenance of the protein, whether the protein remains in its, conformation, and then the degradation of the protein. So these three comprise of the main proteostasis machinery. So we'll see how proteostasis is regulated. We'll come to ask some questions like whether the protein synthesis levels they change with age, whether the components of the translation apparatus are affected, whether there are some errors in protein synthesis also when we age, or is there some age dependent regulation of protein synthesis and the level of translation. So we'll ask these kind of questions. So let's see what we have. So this is how the proteostasis works. In this case, we have the initiation, elongation. This is the ribosome that you can see. So initiation, elongation, and then termination. So that is how the proteins are synthesized, and then later they are sometimes recycled also. So whatever you see in the green here, that happens due to aging, right? Like the elongation factor concentration and activity is reduced with aging. The translation fidelity, that means whether there will be a correct translation happening, that also sometimes gets reduced. There are some studies on that. And 3' UTR translations happens at when you age. Sometimes, the proteins that do not stop where it is supposed to stop. And even in the initiation stage, the ribosome abundance, it decreases, the gene copy number, it decreases, initiation rate decreases. So a lot of these problems happen. So while if we have if we have an increased lifespan somehow, then the translation fidelity, rRNA methylation, these kind of things increase. So these are all, this is just a cartoon that explains everything together. So protein synthesis is one of the most energy consuming processes that we have. It requires almost 75% of the cell's energy. And so what happens? There is reduction of mRNA translation, mRNA translation increases overall energy availability. So let's say mRNA is not translated, then there is more energy left to spend. So it may allow diversion of critical resources that can be mobilized towards cellular maintenance and repair processes, thus promoting organism longevity. Additionally, the slowdown of growth rates and consequently the rates of protein synthesis increases the accuracy of translation and therefore proteome quality and stability. So uh, here what we are discussing is that protein synthesis in itself is a very energy consuming process, and it is very critical for the survival of the cell. Accuracy of translation is important. Rate of protein synthesis is important. If the protein is synthesized too fast, there might be more errors and there would be misfolding. If the protein is synthesized, so it has to be synthesized at a pace where it folds properly. Then the accuracy is maintained, the stability is maintained. These all things are very important. And so this theory is supported by the fact that depletion of ribosomal proteins and translational factors in several model organisms decreases protein synthesis and increases stress resistance and extends lifespan. So essentially, it's kind of like it's kind of like a balance. So initially, when you are young, your most of your energy is spent on protein synthesis and That's where your energy is spent. Mostly you synthesize proteins, you synthesize them properly. But when you age, you have the DNA damage machinery that comes into play, the DNA repair machinery, essentially. Those things, they consume some amount of energy. Hence, your protein synthesis fidelity is, compromised to some extent because of this reason. Is that clear to everybody?
Sir, can you
Yes,
please
sir.
repeat this part? Like several model organisms decreases protein synthesis and increases stress resistance and extends lifespan. Uh, this part I don't understand.
So in that cases, whenever you let's say if there is sporulation, in some cases, so we have such kind of situation. We have animals that hibernate. So in any kind of stress, even there is biofilm formation in case of bacteria when there is any stress given to them. You know these things.
Yes, sir.
So the first thing that they do is to reduce the protein synthesis because that consumes a lot of energy. So if you stop that process, you can use that energy in some other responses. So it's like, it's kind of a budget that you're trying to balance.
Yes,
It's
sir.
kind of your understanding. So the energy is limited. So they use that energy in other kind of situation, in stress response in specific. So yeah, if you give
Yes,
any
sir.
kind of stress to the cell, the protein synthesis is the one which gets affected. So translation, it occurs at a rate of 6 amino acids per second. Twofold decrease in elongation rate is observed in aged rat tissues relative to youthful counterparts. What does this mean? That what happens with aging?
The rate of translation decreases.
Rate of translation, we could also say the translation fidelity.
Rate of
That also,
translation.
yeah, that decreases. So, this organism, this animal,
Mole rat.
yeah,
Naked
it's
mole rat.
naked mole rat.
It's a
Yes.
mole.
Mole.
They are actually outliers of their family of rodents. So usually a rodent lives up to, let's say mice, they live up to a maximum of 2 years. Rat maybe 3 to 5 years. How long do naked mole rats live? Does anybody know?
So I forgot
No,
the
sir.
exact age, but remember that they have the gene P27, which restrict the property of their losing contact inhibition. That's why they don't undergo that cancer in exposure to this radioactive particles.
That is correct. That's one way of it. We are not going to study that. But anyway, naked mole rats, they live. They have a recorded lifespan of more than 30 years. So that's like a tenfold increase in lifespan. So definitely, there's something that we need to figure out what these mice, what these rats are doing that make them live so long. I mean, compared to other rodents. So NMR, like NMR is basically, in this case, it's naked mole rats. Their fibroblasts have fourfold lower rate of amino acid misincorporation. That means error rate is very low than the primary mice. So we are going to study a little bit about this. So in a study, authors
Sir,
had
could you
used,
please show the previous slide once?
yes, yes,
Um, so here the elongation, it's only a twofold decrease in elongation that causes the aging. What is the meaning of twofold?
twice 2x.
Twice two.
Half, I mean, it's like, let's say X is the elongation rate, X by 2 will be the elongation rate in aged cases. So that is a twofold decrease.
Okay, thank you, sir.
Then few authors to study whether translational accuracy has something to do with, aging. So what they did, they used an paramomycin. It's an aminoglycoside antibiotic. So it decreases the translational efficiency, it decreases its accuracy essentially. So the ribosomes which were isolated from the livers of old rats, they had increased sensitivity to this antibiotic. So if when they were treated with this antibiotic, they made 9% more errors than the ribosomes that were obtained from the young rats. So clearly, once your ribosome machinery, it also gets aged, or in the aged cell, the ribosome machinery is more sensitive to making errors. So that's essentially the entire point of this hallmark. So the proteostasis machinery is altered. So the intracellular distribution of newly synthesized proteins. So once these proteins are synthesized, they need to be distributed at the right places. So aging, it does not only affect the translation machinery, but also the ability to control where the protein needs to go. That also is affected due to aging. So there is, again, an example is given. It could be a decrease in availability of nascent polypeptide associated complex due to partial aggregation during aging, and it may result in mistargeting of co-translationary. Yes, Chinjan.
So what's the difference between proteostasis and homeostasis?
Homeostasis is the maintenance of, in general, proteostasis only refers to the proteome, whereas homeostasis refers to even metabolism, everything else uh, in your body, whatever is going on.
It
Mostly, I think
includes the
metabolism.
protein. Like in homeostasis, does it include protein?
Is a good question. I might have to look into it, but I think yes, it definitely does, but it is not specific to protein as such. I mean, if the
Okay.
homeostasis is altered, definitely proteostasis will also have an effect. That's for sure.
So in
So
aging, which is affected first, like the proteostasis mechanism or the homeostasis?
homeostasis is definitely affected in some way or the other, but the thing is, proteostasis is one of the hallmarks. I mean, loss of proteostasis, that is one of the hallmarks, the sign, one of the sign that you are aging. So when we are comparing, so see, I don't know whether you saw the podcast that I sent you yesterday. So Dr. David Sinclair, he is one of the the big shots in this field, one of the greatest person who's living right now, who is working on aging research. So uh, what he tried to say is aging as we see right now is not a disease, but something, it's a fate that you have to suffer. And like 80 to 90% of the people, they have aging. Everybody ages. That's the point. But uh, that's why we do not treat it as a disease. So when you're talking about disruption in homeostasis, we generally refer to some diseased condition, right? But aging in itself, it's also a condition. It also leads to diseases. So yes, aging also causes some effect on the homeostasis of an organism. That is correct. Does that make any sense or not?
Yes. Like,
So anyway, let's go forward. Let's move forward. What do we have? So this is how a protein is organized. So you know, it's there is primary structure, secondary structure, tertiary structure, and quaternary structures. These are important. How the protein, the different levels of protein organization. So how do these happen? Let's see. So the cellular proteome, it is highly dynamic, and these proteins, they undergo major conformational changes for their maturation, for their mobilization, for assembly into different function units, for their function, maturation, and localization. For these reasons, protein need their conformation to be correct. So a percentage of de novo synthesized cytosolic proteins, they fold spontaneously. Others, they require the assistance of chaperones. So you have heard about these chaperones, molecular chaperones? How do they help? I mean, what's their role?
They are involved in like proper folding of the protein structure
That is
during
correct.
post-translational modification.
That is correct. So essentially, that is what is written. So most of the synthesized proteins, I mean, a small part of them, they do not require these, chaperones, but I mean, most of them do. So we will see how a chaperone works. So chaperones, they mostly have, they are also known as heat shock proteins because they serve both as the sensors of alteration protein quality and also as active or passive facilitators of protein folding. Heat shock proteins, they are known because they were identified initially as one of the stress molecules when heat shock was introduced. So that's why the name comes up like that. So this is how a chaperone works. So when this is a chaperone. So when a nascent polypeptide is synthesized, it needs to be, wait, did I? Okay, once it is synthesized, it needs to be folded properly and it forms a mature, protein. If there is some sort of damage, then the chaperone cannot fold this protein properly and this protein gets aggregated. Any example of any disease that you can think of where this protein accumulation happens? Are people following or it's getting difficult
Yes,
again?
following. Yes, it's uh,
Sir, Huntington's.
Huntington's correct. That is correct.
Sir, and also can be called like blood cancer in this because that also involves the like accumulation of WBCs as well.
So
So
blood leukemia is a little bit different actually. It's not accumulation as such. Or
to protein like since
any
I mean,
diseases related to protein accumulation in blood.
in blood, not necessarily. I mean, yeah, any other disease. I mean, blood cancer could be due to a lot of reasons.
Alzheimer's.
That is correct. Absolutely. Alzheimer's and Parkinson's. So what is
So
the protein that is?
Parkinson's basically involves the dopamine thing. Like dopamine is not secreted. That's why loss of protein occurs.
It affects the dopaminergistic neurons, firstly.
Yeah.
So dopamine treatment sometimes helps. But what essentially happens in Parkinson's, we'll be studying about it in the next session, is accumulation of a protein called alpha-synuclein in the substantia nigra of the brain that happens again due to the loss of proteostasis. In case of Alzheimer's, it is amyloid beta which gets aggregated. All these are proteins. So again, these diseases are something that happens, even Huntington's, as a matter of fact, it happens at a late onset. I mean, once you age. So if you even leave Huntington's out of this picture, at least Parkinson's and Alzheimer's, they always happen once you age. So definitely, loss of proteostasis, proteostatic machinery plays a good role in aging, right? That we can say. So let's see what do we have next. So, these chaperones, they have two distinctive regions. One is the substrate binding region, and another one is the nucleotide binding region where ATP is bound because sometimes chaperone they utilize ATP for the folding of the protein. So it's sometimes energy dependent. So in the context of aging, we have seen that the content of chaperones uh, they have changed with the age that has happened. So there is decreased transcription protein levels of HSP70. HSP70 is a molecular chaperone. and it has been described in both senescent cells in culture and in many tissues from old organisms. So also there is reduced availability of ATP once you age. So hence the chaperone effect activity is also lowered once you age. So see, at every point we are seeing that with age there is a change in protein synthesis that gets lowered. Protein folding maturation that has an effect, and we will see that protein degradation also is affected with age. So does that make any sense? We will see what
Yes, sir.
happens. Chaperones in sorry. So chaperones are in the organelle response to stress. So quality control of organelle resident proteins is essential for proper organelle function. So the levels of several ER chaperones have been shown to decrease with age, as we said, and in various tissues in rodents and in senescent human fibroblast in culture. For some of the chaperones, addition to drop in protein levels, a decrease in their activity with age has also been reported. So that is for chaperones that we have discussed. Now let's see a protein life cycle. So initially it will be synthesized, right? Then there will be co-translational processes. So in the time of synthesis, what could go wrong? There could be deregulated stoichiometry of translation components. Essentially, all the translation components are not available. The mRNA is sequestered, or there is decrease in overall translation rate. In co-translational processes, there could be misfolding or mislocalization. During its functional lifespan, also there could be, problems which could happen, and during degradation also there could be problems due to aging. So one of it you have already studied, autophagy. I mean, if you have studied about it, autophagy is also kind of protein degradation. I mean, it degrades a lot of things, protein being one of the main components. So we'll talk about it. We'll talk about degradation. This is how the life of protein is. So first three panels, they show you how the proteins are synthesized, then they are refolded or repaired and then degraded. If it doesn't degrade, then it gets aggregated. So even in, as I said, it's a hallmark. So a folded protein uh, is usually, if it gets unfolded, if it is non-functional, then it has these machineries. So one is chaperone mediated autophagy is there. So you have studied about autophagy. Then there is proteasomal degradation, or the protein could be again refolded so that it can be reused. But if it gets aggregated, then there is it's a sign of aging. So let's see what happens. So abnormal proteins, then they are degraded in the cells. So what are these abnormal proteins? They could be incomplete proteins, missense proteins. There are excess subunits, damaged proteins, or misfolded proteins. So let's see what happens. Protein degradation happens mostly through the ubiquitin proteasome pathway for at least intracellular proteins. The rest 10 to 20%, they are degraded through the lysosomal process, that is autophagy. You have studied about this. I mean, read about this. And what happens? You have heard about ubiquitin proteasome pathway? Anybody?
Is it that oxidative phosphorylation thing?
No, it's
Then
protein degradation.
not heard.
So how are proteins degraded? For this, people got Nobel Prize in 2004. These are the three people who discovered this system. So let's talk about ubiquitin. You must have heard about ubiquitin.
Yes, sir.
So ubiquitin, it's a small peptide. It acts as a tag. It's of 76 amino acid. The C-terminal glycine of the ubiquitin interacts with the lysine of the protein. So ubiquitin can be attached as monoubiquitins, like only one ubiquitin getting attached, or polyubiquitins. So first of all, what is ubiquitination? What kind of
It is
modification is it?
post-translational modification.
That is post-translational modification. You're correct. So it's a post-translational modification, and these ubiquitination, usually if you have studied about it, they function as signals of how the protein is supposed, probably signals for localization, where the protein needs to go. Sometimes it also signals for damage repair and other machineries. So ubiquitination is important in that sense. And then we will talk about the steps involved. The first step is ubiquitination of the protein. Second is recognition of this ubiquitinated protein, its degradation, and then deubiquitination. Let's see how it happens. So uh, in a simplified diagram, so a target protein is ubiquitinated using ubiquitin enzymes E1, E2, and E3. We'll talk about these three enzymes. And then once it is ubiquitinated, it is detected by the proteasome machinery. This is how the proteasome looks like on your right. It causes proteolysis, degradation of the protein. So in this ubiquitin, enzymes, conjugating enzymes, we have the E1 ubiquitin activating enzyme, then the conjugating enzyme, and the protein ligases. So the E1 acts first, then there is conjugating enzyme, which carries this ubiquitin moiety, and then there is the ubiquitin protein ligases, which are specific to specific proteins. So that's why they are high in number. So they will take the ubiquitin from E2 and they will place it on the protein, on your target protein. So if it's like this, if you see the previous diagram, so E1, basically ubiquitin attaches into E2, E3 will help, this ubiquitin to relay from E2 to the target. So that is the function here of these three. So this ubiquitin proteasome system, uh, its gene is comprises at least 5% of the genome. That's pretty much high. I mean, it's a lot of genes are functioning. So E1, there are one or two activating enzymes. For E2, there are 10 to 20 conjugating enzymes, and E3, the ubiquitin ligase, there are 500 to 800 E3s. And then there are deubiquitinases, which remove this ubiquitin. So there are like 100 specific proteases in this respect. So again, ubiquitination, as I said, monoubiquitination could be there or polyubiquitination. So monoubiquitination is usually it signals for receptor internalization or endocytosis or transcription regulation. Whereas polyubiquitination is essentially it means that the protein is going for proteasomal degradation or DNA repair. So if there is polyubiquitination in lysine 11 or lysine 48, it signals for UPS mediated proteolysis, ubiquitin proteasome system. So this is how the proteasome looks like. It has two components, the 20S part. This is the cylindrical part. So if you see, it's something like this. The cylindrical part, this has mostly all the enzymes for proteolysis. Then there is something called something like a cap in both the ends, right? They are present in both the ends. These are the 19S particles. So what would happen? A protein would come. This will sense the ubiquitinated protein. Then the protein will be degraded in this machinery. Which way is the way it enters and which way it comes out? I mean, such kind of studies are have been done, but it's not clear. But this is how it looks like. So this cylindrical part, the 20S proteasome, it contains your proteases. So caspase-like proteases, trypsin-like proteases, chymotrypsin-like proteases. So what will be the function of these proteases?
They will hydrolyze the peptide bonds between the amino acids.
That is correct. So essentially it will break down the proteins, right?
Yes, sir.
So that is essentially what happens then. So the catalytic activity is not in all beta subunits, but some of them. And these are members of the N-terminal nucleophilic hydrolase superfamily. So there are three site specificities, as I said, chymotrypsin-like, which target the non-polar and aromatic amino acids, trypsin-like for polar and basic amino acid, and caspase-like for glutamate, essentially. So then there is deubiquitination. So once this, ubiquitin, the role of ubiquitin is used, I mean, it's done, then this needs to be deubiquitinated so that it can be reused for other processes. So what happens? What could happen here? Let's say if there is aggregation.
So where will the aggregation might occur?
Proteins are getting aggregated. Lot of
Yes,
proteins
sir.
are getting aggregated. What might happen?
Then the proteasome will not be able to like degrade it since of its because of its bulk size. So
Yes, that is correct. So this entry will get jammed, no?
jammed.
Most of this time, this is exactly what happens. So they stop functioning in that sense. So initially, what could happen? The dysfunction could be decrease in proteasome subunits. That makes sense. There could be some alteration in these subunits. There could be disassembly of proteasomes. Proteasomes in itself are getting, disassembled because of this aggregation, or they could be even inactivated by these protein aggregates. So they are stopping the entry of protein. They are completely, uh, the entire system has stopped. So again, this decline in proteasome function during aging and senescence, that has also been observed in several mammalian tissues. So the examples have been given here. So what else do we have? Okay. So if we treat fibroblast with proteasome inhibitors, they have shortened replicative lifespan and senescent-like phenotype. Where would we expect something like this to work out? Proteasome inhibitors in which kind of cells? If I want to use it as a treatment strategy, anybody?
Liver.
I want to reduce the lifespan of a particular kind of cell. Which kind of cell I would like to reduce the lifespan of?
Neurons.
Neurons? No, no, not neurons. If I reduce the lifespan of neurons, it will die. No, what's the point?
So what's the question?
So proteasome inhibitors have been used in cell culture models, and they have seen that if they inhibit the proteasome function, then the cells have a reduced lifespan. So which kind of
Yes,
cells
sir.
would I want? Yeah, which kind of cells would I like to reduce the lifespan of? Or I would like to kill these kind of cells basically.
Phagocytic cells. I mean pathogenic cells. Sorry.
Pathogenic cells, yes, but again, these are not very specific. Maybe cancer cells.
Yes, sir.
So cancer also proteasome inhibitors are used as a method of therapy. So see, all these, all the while we are discussing about aging, it's very, very closely related with cancer. So then we,
How is it used?
how is it used
Like
in a sense?
how can we insert it at the since
For
it's cancer cell,
delivery
it's
of
already has reduced the property, right?
cancer cells.
It's we want to kill the cancer cells, so how can we use this?
So for drug delivery approaches, that is a separate topic we will discuss. I mean, it's like we can use nanoparticles, we can use other methods of delivery. Sometimes there are, the conditions of
Genetically
the
engineered bacteria.
yeah, engineered bacterias or vehicular modes, other kind of vehicular modes are also used. Sometimes the conditions of these cancer cells, they make us, devise certain strategies where, let's say, if the pH is lowered, there only these drugs will be active, something like that. So
Okay.
that's how we make the specificity. But anyway, it's again in an experimental stage as of now. So then there is a question whether the proteasome is the proteasome equally affected in all the cell types, right? So, or whether the effect on these of this proteasome machinery is different in different cell types. So when this kind of a study was done on C. elegans, it was observed that the dorsorectal neurons, they exhibit more decline in proteasome activity than the muscle cells. So again, the level of loss of proteostasis could differ from the cell type. So you know what ESCs are? Embryonic stem cell. Yes, so they do not usually undergo replicative senescence and they are considered to be immortal in culture. So when they're immortal, we are supposed to think that they are immortal because they can maintain their protein properly. So there might be some mechanisms how they maintain their proteome and they avoid the senescence. So when they studied human embryonic stem cell, they saw that there was high proteasome activity compared to differentiated counterparts like neurons and fibroblasts, etc. So that's also we talked about the main components of proteostasis, the synthesis of protein which gets affected due to aging because the synthesis is sometimes it's more error prone. The synthesis gets lowered. So these kind of problems happen with aging. Then secondly, we discussed about the maintenance, the conformation and maintenance. So the molecular chaperones, they reduce in number. Sometimes they do not fold properly. It is an ATP dependent process most of the time. That's why ATP is no longer available. So again, protein folding is affected with aging. And then we said that protein degradation is also affected with aging. So all the parts of proteostasis is affected with aging. That's why we call it as a hallmark that loss of proteostasis is one hallmark for aging. Any more questions?
No, sir.
No, sir.
Is it clear or is it getting difficult again?
That's clear.
Sir, could you please explain the human ESCs once more?
See, so what we talked about that proteasome, if you inhibit this proteasome, then the lifespan of the cell it decreases significantly. Correct? That is clear to you?
Yes, sir. Yes.
Fine. Now these embryonic stem cells, the stem cells, the role, I mean the good thing about them. Let me just stop the recording here.